Good morning, everyone. I am Ms. Republica Borgohai, Assistant Professor, Assam Downtown University, Guwahati. So today I will be discussing about what is the maple syrup urine disease. So in my previous videos, I just discussed regarding the introduction part of amino acid and the classification of amino acid. So in case of maple syrup urine disease, the degradation of branched chain amino acids, it involves the branched chain alpha keto acid dehydrogenase, that is BCKDH. A deficiency of this complex leads to a buildup of the branched chain amino acid, that is leucine, isoleucine and valine, and their toxicity or their toxic byproducts in the blood and urine, giving the condition the name maple syrup urine disease. And now comes the standard amino acids. So there are more than 300 amino acids, and only 22 form all proteins in plants as well as animals. Humans can produce 10 of the 20 amino acids. The others must be supplied in the food. Failure to obtain enough of even one of the essential amino acids, those that we cannot make, results in degradation of the body's proteins. So we know that 22 essential amino acids, they form all proteins in plants as well as in animals. So humans can produce 10 out of the 20 amino acids. The others, they must be supplied in the food. And if there is a failure enough to even obtain one essential amino acid, then there may be some degradation in the body's proteins, in the intake of body's proteins. So now comes the nutritional classification. So according to the classification based on nutrition, they are divided into three types. So they are, number one, essential amino acids, those that we cannot make, results in degradation of the body's proteins. Secondly, it is conditional amino acids, and thirdly, it is non-essential amino acids. So now comes the essential amino acids. So the amino acids that are to be supplied through diet are called as essential amino acids. They cannot be produced by the body itself. The essential amino acids are valine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine and tryptophan. Now, what are non-essential amino acids? The amino acids that can be synthesized by the body are called non-essential amino acids. They are glycine, alanine, serine, cysteine, aspartic acid, glutamic acid, asparagine, glutamine and proline. Now, conditionally essential amino acids. So these are essential only in certain cases. So some amino acids like arginine and histidine are growth promoting factors. They are not synthesized in sufficient amount during growth. So essential in growing children, pregnancy and lactation. Tyrosine is produced from phenylalanine. So if the diet is deficient in phenylalanine, tyrosine will be required as well. So as tyrosine is produced from phenylalanine, so if the diet is deficient in phenylalanine, so tyrosine will be required as well. So now the non-standard amino acids. Aside from the 22 standard amino acids, there are many other amino acids that are called non-proteinogenic or non-standard. Those either are not found in proteins, for example, carnitine, GABA, or are not produced directly and in isolation by standard cellular machinery, for example, hydroxyproline and selenomethionine. Non-standard amino acids that are found in proteins are formed by post-translational modification, which is modification after translation during protein synthesis. These modifications are often essential for the function or regulation of a protein. For example, the carboxylation of glutamate allows for beta binding of calcium cations, and the hydroxylation of proline is critical for maintaining connective tissues. Some non-steroid amino acids, they are not found in proteins. For example, they include lanthionine, 2-amino isobutyric acid, dehydroalanine, and the neurotransmitter gamma-aminobutyric acid. Non-steroid amino acids, they often occur as intermediates in the metabolic pathways for standard amino acids. For example, ornithine and citrulline occur in the urea cycle, part of amino acid catabolism. So now comes the discussion for Zwitterions. So what are Zwitterions? The amine and carboxylic acid functional groups found in amino acids allow them to have amphiprotic properties. So carboxylic acid groups, that is CO2H, can be deprotonated to become negative carboxylates, CO2, and alpha amino groups, that is NH2, can be protonated to become positive alpha ammonium groups, that is NH3. So here, in case of Zwitterions, the amine and the carboxylic functional groups, they are found in amino acids. They allow them to have amphiprotic properties. That means the carboxylic group, that is CO2H, it can be deprotonated to become negative carboxylate, that is minus CO2. And alpha amino group, that is NH2, can be protonated to become positive alpha ammonium group, that is plus NH3. So these are the non-ionic form and the deuterionic form, which I have explained before. So now comes what is isomerism. Of the standard alpha amino acids, all but glycine can exist in either of two optical isomers called L or D amino acids, which are the mirror images of each other. While L amino acids represent all of the amino acids found in proteins during translation in the ribosome, and D amino acids are found in some proteins produced by post-translational modifications. They are also abundant components of the peptidoglycan cell walls of bacteria, and D-serine may act as a neurotransmitter in the brain. So what is isoelectric point? At pH values between the two pKa values, the Zwitterion predominates, but coexists in dynamic equilibrium with small amounts of net negative and net positive ions. At the exact midpoint between the two pKa values, the trace amount of the net negative and trace of net positive ions exactly balance, so that average net charge of all forms present is 0. This pH is known as the isoelectric point. So now discussing about the stereoisomerism. Stereoisomers are isomeric molecules that have the same molecular formula and sequence of bonded atoms, that is constitution, but differ only in the three-dimensional orientations of their atoms in space. This contrasts with structural isomers, which share the same molecular formula, but the bond connections and or their order differs between different atoms or groups. So this was the discussion regarding the stereoisomerism and isomerism, and lastly I want to conclude it with the functions of non-protein functions. That is, in humans, the non-protein amino acids, they also have important roles as metabolic intermediates, such as in the biosynthesis of the neurotransmitter gamma-aminobutyric acid. Many amino acids are used to synthesize other molecules. For example, tryptophan, it is a precursor of the neurotransmitter serotonin. Tyrosine, it's a precursor of the neurotransmitter dopamine, and glycine, it's a precursor of porphyrins such as heme. So arginine, it's a precursor of nitric oxide. Ornithine and S-adenosyl methionine are precursors of polyamines, and such as phenylalanine and aspartate and glycine. So phenylalanine is a precursor of various phenylpropanoids, which are important in plant metabolism. So here I conclude my topic regarding the amino acid. So in my next video I will be discussing regarding the protein, its introduction, its classification, and the various divisions. Thank you.